So when you go through this universal codon table, you may also want to understand that there are more than one codon coding for a single amino acid that's referred to as a degeneracy. And also, if there is a mutation that changes one base to another base, say for example GGG to GGC, it may not result in any change in protein synthesis or code for the same amino acid, glycine. But sometimes they would change the amino acid itself. That is called as a missense mutation. If it doesn't make any change, that is a silent mutation, but sometimes it can result in a stop codon being introduced instead of actual codon. So that is called as a nonsense mutation. So in some cases, there could be a deletion of one base in the coding region of the DNA, resulting in, yes, one base deletion in the mRNA where an amino acid is supposed to go. That will result in possibly a frame shift mutation where the whole reading frame of the codon is shifted, or it may result in stopping the entire protein synthesis there, resulting in a shorter protein, or it may extend the protein to become a longer protein. Okay? So all these are possible in the case of a deletion or insertion mutation. So understand how these things work. The next, we are going to get into the details of protein synthesis itself. So protein synthesis can be divided into three major stages. The first stage being initiation, the starting of protein synthesis. The second stage being elongation, extension of the protein. The last stage is termination, stopping of the protein synthesis. So in the initiation stage, when the mRNA that comes out in the cytoplasm, after all the processing is done, in the case of eukaryotes, the ribosomes recognize a region near the start codon, which is a translational start codon, and bind, and that is the beginning of protein synthesis. In the initiation, there's also a methionine-containing tRNA. There is always a start codon, is a methionine codon. It brings in the methionine amino acid. Sometimes it is a modified methionine. It binds to that P site of the ribosome. There is a large subunit of the ribosome also comes and bind. Initiation factors, GTP, are used. In the second stage of protein synthesis is called elongation. Elongation is where more and more amino acids are being added to the elongating protein, and that is brought in by the tRNA. So in this assembly of large subunit, small subunit, a new tRNA will come in. It's called as a codon recognition, and a peptide bond is formed between the existing amino acid and incoming amino acid, and that is formed by the ribosome itself. The mRNA and the tRNA and the protein complex of the ribosome perform this peptidyl transferase activity. There's no separate enzyme involved. And once this is done, the entire tRNA that was in A site will move to the P site, and we will see all the details later on in the lecture. But once that translocation happens, the protein synthesis continues all the way until it ends and at the stop codon. So there are three different stop codons. One of that is recognized by the release factor, and that release factor will come now release this ribosome, and then it will also release a protein, and they will go on to make more proteins. The ribosomes will go on to make more proteins. The mRNA can be read by other ribosomes. So in this process, it happens in initiation, elongation, and termination. Once a protein is made, it's not all said and done. There are some proteins need to be modified. Either the amino terminus part of the protein is removed, it's called as zymogen activation, or the protein is now modified, embedded into the endoplasmic reticulum or other membrane, and it is the signal peptide that comes in, recognizes endoplasmic reticulum is removed, or the protein is glycosylated by adding an oligosaccharide, or it can be added a lipid group to make a lipoprotein. It's called myristylation process. All these are considered as a protein processing. So the protein synthesis is an important process. That is what makes the different enzymes and different transport protein, different structural protein in the body. But it starts all the way at the nucleus, the gene being expressed to make the RNA. And in eukaryotes, the RNA is processed further in the nucleus. comes out in the cytoplasm. Now the ribosomes bind to that mRNA, and then it makes the protein, and the proteins are processed. In prokaryotes, the life is simple. It's short and simple. And in that case, the prokaryotic genes are organized in units called operons. We are going to see that in the next chapter. And they are expressed, transcribed and translated in the same place at the same time. Only in eukaryotes, transcription and RNA processing happens in nucleus, translation happens in the cytoplasm. And there are a lot more steps involved in RNA processing and the protein processing. So this is a kind of a summary of this chapter of transcription and translation. And the next chapter, we are going to look at how the genes are controlled or how the expression of the gene is regulated.